Cell evolution. When biomolecules began to form in the ocean around 4 billion years ago during early Earth conditions, cells are theorized to have formed through a process called abiogenesis. Around, that should actually say 3.8 billion years ago. So to explain that sentence, the cell theory is that all cells come from existing cells. Well, what about the first cell? Well, we think that the first cell arose in Earth's early ocean billions of years ago through this process where biomolecules that you learned about last unit, carbohydrates, lipids, proteins, and nucleic acids, and the building blocks for those biomolecules, they're monomers, like for example, amino acids and nucleotides, fatty acid chains, glycerol. Those molecules are always built and used inside of cells, but the idea is that these molecules actually formed on their own before cells existed. And over millions of years of trial and error, these molecules came together to form the first cell, which became the ancestor to all life on Earth. And this is a diagram of the Miller-Urey experiment that supports that theory. This experiment simulated Earth's early water cycle, and the molecules that are theorized to have been around at that time, like for example, methane, ammonia, hydrogen gas, and water, which contain the elements which, if they react with each other in the right way, they could actually form some of these monomers, like for example, amino acids or fatty acids. And they simulated Earth's early ocean and how the water would evaporate from the ocean. There's storms, electrical storms, precipitation going back into the ocean. And they simulated that, and they had this collecting chamber. And in this collecting chamber, they eventually started seeing amino acids, which we know are the precursors to proteins, as well as fatty acid chains. And that supported that this could have happened in Earth's early ocean. These biomolecules also are theorized to have been brought over from meteorites, for example. But either way, once the biomolecules were available, cells started to form, and then cells then isolate the reactions necessary to build more of these biomolecules. And now these biomolecules are only built and used inside of cells. So these very primitive cells that formed billions of years ago were small, basic, and lacked internal organelles. And they look similar to modern-day bacteria, which are still the most abundant life forms. As modern-day bacteria, as well as these cells, are prokaryotic, meaning they lack internal membranes. Internal membranes, another word for that is organelles. Inside of their cells, they're unicellular. However, even modern-day prokaryotes are more complex than these first cells. These first cells passed on their characteristics to all life today and can be seen as common features to all cells. So the common features to all cells is a cell membrane, cytoplasm, genetic material, and then I should have had you write this one, ribosomes. So these are the 4 structures seen in all cells today because the ancestor cell had to have these 4 things to be a functional cell. And kind of redundant, but it's also right here as well. So the most basic of cells must contain these parts in order to be a cell. So both prokaryotes and eukaryotes are going to have these 4 cell parts. So the cell membrane, which is also called the plasma membrane, and it's going to be a waterproof barrier between the cell's internal environment and its external environment, and it's going to regulate molecule entrance and exit. And if you were to zoom in on the cell membrane, this is the phospholipid bilayer. made of phospholipids, where their fatty acid tails are facing each other and their polar heads are facing the water inside the cell and the water outside of the cell. So it creates this waterproof barrier separating the water inside of the cell from the water outside of the cell. We have the cytoplasm, which is the water-based fluid called cytosol, C-Y-T-O-S-O-L, and it's right here. Jelly-like fluid made mostly of water and dissolved solutes, and it forms the base of the cell's chemical reactions and molecule transport. So remember, water is a universal solvent, so therefore cells are water-based in order for things to dissolve and react in. And then we have our genetic material, which if you zoom in on this scribble is going to be DNA, genetic codes for synthesizing proteins. And proteins are necessary to allow the cell to function and to reproduce. And so where the proteins are actually made is in ribosomes, small structures made of RNA and proteins that are the sites of protein synthesis. So this is, for example, a polypeptide coming out of the ribosome. And a ribosome is a tiny little structure, so a cell can have thousands of ribosomes. And then what's being read through the ribosome is the mRNA going through the ribosome. We won't focus on that now, though. Okay, so cell evolution process. So first of all, we have inner membrane folding. Due to genetic mutations, cell membranes began to pinch and create many internal membranes inside the cell. These created little pockets in the cell that isolated specific chemicals and reactions. These internal membranes are now called organelles. Descendants of this mutated cell with organelles inherited this trait. Organelles increase the efficiency of cells as they compartmentalize cell functions. This allows cells to grow much larger. Cells with organelles are called eukaryotes. Examples of organelles are the nucleus, where DNA is replicated and transcribed, lysosomes, where hydrolysis or digestion reactions occur, and the endoplasmic reticulum. Reticulum, but it's also just called the ER. And this created what's called the endomembrane system in eukaryotes. So I'm going to go to this diagram right here. So we're starting off with an ancestral prokaryote that existed billions of years ago. And one of these prokaryotes that had the basic cell structures in its genetic material had a mutation, and that mutation led to its cell membrane being folded incorrectly, where these little like internal cell membrane pieces were also being created, these little pockets or infolding of the plasma membrane. So this is evolution event 1, and this internal pockets, they would isolate specific reactions, whatever chemicals they happen to isolate. And so that created the nucleus, which isolated the DNA, for example, as well as the endoplasmic reticulum and the lysosome, like I said. Vesicles and vacuoles were created this way. So this created a primitive eukaryote. So cell with nucleus and endomembrane system would be called a primitive eukaryote. So then we have the theory of endosymbiosis. Endosymbiosis. So I'll read all this first and then explain it on the diagram. Some prokaryotes had evolved the ability to use oxygen during a process called aerobic cellular respiration. which is a series of chemical reactions that breaks down biomolecules such as glucose in order to generate energy needed to power the cell. Using oxygen in this process generates a lot more cellular energy, which we're going to write in parentheses ATP. So the theory is that some of these prokaryotes were engulfed by a larger primitive eukaryote, and instead of being digested, it became an organelle within the host cell whose function was to generate cellular energy. And the name of this organelle is now called a mitochondria. Oh, I should say mitochondrion. And then plural is mitochondria. All subsequent cells from this host cell have mitochondria due to the replication of these organelles before cell division. So let me explain that better. So in Earth's early ocean, we had our prokaryotes, simple smaller cells, and then we had our primitive eukaryotes that had a nucleus. And because they could isolate and compartmentalize cellular reactions, they were able to grow larger. Even though their surface area to volume ratio is smaller, having organelles, each of those organelles has a smaller surface area to volume ratio, and so therefore they still are efficient. And so these larger cells were eating the bacterial cells, the smaller cells, and then digesting the bacteria in their lysosomes. However, in this second event right here, bacteria that had the ability of using oxygen in order to create ATP was engulfed by a primitive eukaryotic cell, but instead of digesting the cell, it retained the cell, and the cell lived endosymbiotically inside of the host cell. And it was a mutualistic relationship because the host cell was able to get a lot of ATP, cellular energy, and then the bacteria was getting a home, a protection, and then the food, the digested food from the primitive eukaryote was given to these bacteria cells to use to make ATP. So it was a happy relationship, and it's a relationship that is still around today. So this bacteria replicates inside of its host cell and makes a bunch of these mitochondria organelles. And when this cell divides, the mitochondria bacteria are passed on to all of the daughter cells as well. So in each of your cells, you have a few hundred to a few thousand mitochondria, and those are actually kind of bacteria that have lost their independence inside of you. So the descendants of this event, event number 1 and then event number 2, are going to be all heterotrophic eukaryotes. So for example, animals like ourselves and fungi have an endomembrane system, and we have a mitochondria. So then for event number 3, so this should be here. So we have event 1, intermembrane folding, and then event 2, endosymbiosis of the mitochondria, and then event 3, later in time, another endosymbiotic event occurred, but with a prokaryotic cell, a bacteria that could photosynthesize, meaning it can make carbohydrates from carbon dioxide and water using light energy. And once the host engulfed and retained this cell, they also lived happily ever after, and it became an organelle we call the chloroplast. And descendants of this event have both mitochondria and chloroplasts. So now let's go back to our diagram. So here is after event 2, we have cells that have a mitochondria as well as the endomembrane organelles. And then our descendants, us, we just have those 2 events. And now we have event 3 right here. One of those descendants engulfed a bacteria that could photosynthesize, which is this green bacteria right here. It has chlorophyll, which enables it to absorb light energy. And then these descendants are the photosynthetic or autotrophic eukaryotes, and they have both the chloroplast, which is going to make carbohydrates and oxygen through photosynthesis, and then they have the mitochondria, which is going to use the carbohydrates and the oxygen to make cellular energy. So evidences for endosymbiosis is chloroplast and mitochondria organelles actually look just like bacteria. So they have their own DNA and they have their own ribosomes. They're going to have cytoplasm that's similar to a bacteria's cytoplasm. They are the same size as typical bacterial cells. And then they replicate independently inside of a host cell, and they do so through binary fission, which is how bacteria replicate. And they have a double membrane, meaning that they have the membrane that they had when they were independent cells, and then the membrane they acquired when they entered the host cell, and a vesicle formed around the host cell made of the cell's membrane. And so that process you're going to learn later this unit, but it's called endocytosis. So they're going to have a double membrane due to that. So all super important. Okay. So why are eukaryotes and prokaryotes both around today? Who's better? So evolutionary increase in cell complexity. Prokaryotic cells are the most primitive of modern day cells. Prokaryotic cells lack a nuclear membrane surrounding the DNA as well as other internal membranes or organelles. Internal membranes or organelles isolate specific reactions to create a more organized and efficient factory. Since they do not have organelles, prokaryotes, prokaryotes do not have organelles, they are less efficient at metabolism and other cellular processes. So to offset this, prokaryotes need to be extremely small. However, prokaryotes have evolved a cell wall for protection and structural support, as well as other variant structures such as pili, which is how bacteria can meet up with each other, cilia, which is how they can gather nutrients from their environment, and I don't know what I'm looking for here, probably a flagellum, just singular for flagella, so it's this tail-like structure right here that enables the bacteria to swim. So bacteria and archaeobacteria are the only organisms to have prokaryotic cells, and they are always unicellular. Eukaryotic cells contain a nucleus. You do contain a nucleus and other internal membranes. Again, organelles. And they are going to increase the efficiency of cells by isolating and compartmentalizing cellular reactions. Organelles allow eukaryotic cells to be much larger than prokaryotic cells. I'm sorry, these notes are a bit redundant. A nucleus is a membrane-enclosed cell compartment that encloses DNA. Other examples of organelles include vesicles, which transport proteins, and mitochondria, which metabolizes energy. All life on Earth, besides bacteria and archaeobacteria, are composed of eukaryotic cells, and this includes plants, animals, protists, and fungi. So now we're going to look at specific organelles. So first of all, we have what's called endomembrane organelles, and that's going to be diagrammed right here. We have the nucleus, which I've talked about a lot. Here's all the information that you need to know about the nucleus. And there's also a region inside the nucleus called a nucleolus, where ribosomes are assembled. So another cell structure, not membrane bound, so not organelle. We're going to have ribosomes here. And these are found in prokaryotes and eukaryotes. And again, this is the site of protein synthesis. And ribosomes in eukaryotic cells are going to be free floating in the cytoplasm, but also along what's called the rough endoplasmic reticulum. These little dots studded along the ER are going to be ribosomes. And this large folded structure right here is the rough endoplasmic reticulum. So that's our next one. So a network of membranous sacs and tubes. Function is to synthesize membranes and compartmentalize the cell to keep proteins formed in the rough ER, separate from those of free ribosome proteins. So the two types of ER are rough and smooth. Rough ER contains ribosomes, so it's going to be rougher in texture, and the smooth ER does not. The smooth ER is going to synthesize lipids and then also detoxify the cell. And then since it synthesizes lipids, for example, phospholipids, you could say this is how they build their cell membrane components. And then we have, after the endoplasmic reticulum, the Golgi. And I'm going to go over the diagram first for it. So here is the smooth and rough ER, and they come out of the nucleus. And when lipids, for example, are synthesized in the smooth ER, and proteins are synthesized in the rough ER, little vesicles are going to form and bud off of the ER, and they are going to transport materials to other organelles. So for example, there's proteins inside of these vesicles, and they are going to bud off. You can see it like budding off of the ER right here, and bring those proteins to the Golgi. And the membrane of the vesicle is going to fuse with the cis face of the Golgi and deliver those proteins to the Golgi. And then in a series of reactions that happen in the Golgi, the proteins that are brought to the Golgi are going to be modified, chemically tagged, and they are going to be packaged into export vesicles outside on the trans region of the Golgi. And these proteins will then either exit the cell, like for example, if they're hormones and they need to be out of the cell, or they'll be taken to, for example, the lysosome if they're enzymes needed for digestion, which is what the lysosome does. So think of the Golgi as like the UPS for proteins. They modify, package, and ship proteins for delivery. And the ER is going to bring the proteins to them in this manner right here. So also contains flattened sacs and has directionality. So the cis face is going to receive vesicles from the ER, and then the trans face is going to send vesicles back out into the cytosol and to other locations, or to the plasma membrane for secretion, which means out of cell. And one thing I want to say before we move on is that the Golgi and the endoplasmic reticulum are both highly folded, which means that they are going to have a high surface area to volume ratio, and this is going to increase the cell's efficiency at producing proteins and lipids and getting them transported. It's going to be a very efficient factory because of all of those folds that are all creating these vesicles at the same time. Lots of proteins can be packaged at once if you have a bunch of flat folds. All right, function of Golgi is all right here. I've already said it. Moving on to lysosomes. So lysosomes are going to be organelles that have hydrolytic enzymes. Hydrolysis means to break with water, and so they are going to hydrolyze biomolecules no longer needed by the cell. So polymers are going to be broken down into monomers. Yes. Plants have a similar organelle to lysosomes called peroxisomes. Similar to lysosomes, and they catalyze reactions that produce hydrogen peroxide. Enzymes in peroxisomes then break down hydrogen peroxide into water. Then we have vacuoles, which are basically large vesicles that stem from the ER and Golgi. So this is where they're made. They bud off of them. And there are food vacuoles for storage of food, contractile vacuoles to expel water, and then a large central permanent vacuole, which is going to be found only in plant cells. And we're going to do a lot of stuff in class with this, I promise. And then for our energy organelles, so not endomembrane organelles, we have the mitochondria, which is plural. Mitochondrion is singular, and this is the site of cellular respiration. And the outer membrane is smooth, but the inner membrane has folds called cristae, and this is going to increase their surface area. And then mitochondrial, what is that? Oh, matrix is enclosed by inner membrane, and it's the location of the Krebs cycle of cellular respiration. And it's also going to be where you can find mitochondrial DNA and their ribosomes. The number of mitochondria in a cell correlates with its metabolic activity. Cells with a high metabolic activity have to have more mitochondria. So for example, cells that move or contract, like muscle cells, are going to have high amounts of mitochondria because they need more cellular energy. Then lastly, we have chloroplasts. And these are going to be where photosynthesis occurs. And then inside, it has the, pigment chlorophyll. And inside of its double membrane, it has thylakoid discs. And then it has stroma. So the thylakoid discs are going to be where the light-dependent reactions of photosynthesis occur, and then the stroma is going to be where the Calvin cycle occurs, and is going to be where the cell parts, like the DNA, the ribosomes, and the enzymes of once the chloroplast used to be a bacteria cell, so it's going to contain those cell parts. So for your visuals, here's a plant cell with all the organelles, animal cell, and then bacteria cell would be like basically this primitive cell that we diagrammed earlier. But we're going to do a lot more about cell structures in class, so we are done with the notes.